Have you ever just stopped to think about the incredible world that's buzzing with activity all around us? I mean, stuff happening at scale so tiny, movement so fast we just can't see them. I'm talking about the nanoscale, where molecules are doing these intricate dances, proteins are folding, cells are just, well, living their complex lives in real time.
Yeah, it's a whole universe of constant, almost frantic motion.
Exactly. But how do scientists even begin to, watch this? Especially when it's all happening so incredibly small and super fast. It always felt like, I don't know, trying to photograph hummingbirds with an old box camera.
Hey, yeah, it's a pretty good analogy, actually. The standard tools have really struggled.
Well, today we're doing a deep dive into something really groundbreaking, a technology that's like totally changing how we see at these tiny scales.
Mhm.
It promises to finally let us watch that microscopic ballet.
Mhm.
Imagine a camera, right? But one that doesn't just take static pictures. Instead, it sees the world more like our brain does, in instantaneous events, processing information on the fly.
It's fascinating stuff.
We're diving into the world of neuromorphic localization microscopy. Sounds complex, but the idea is actually quite elegant.
It really is. And our discussion today, it centers on a really important article published in Nature Nanotechnology. The title is Achieving Nanoscale Precision Using Neuromorphic Localization Microscopy.
Yeah.
It's by Rohit Mangal-Wedekar and a whole team of researchers. And this paper, it introduces us to a completely new kind of sensor.
Not just better, but different.
Exactly. Not just tweaking the old stuff. This is a fundamental shift. Genuinely inspired by how our brains work, it's really pushing the edges of what we can even observe.
Okay, so for you listening, our mission today is to really unpack how these brain-inspired cameras actually tick. How do they work? We'll get into how they manage to overcome the really stubborn problems that traditional imaging has faced for, well, forever, and why getting below 20 nanometer precision with millisecond timing is such a massive game changer.
It's not just about seeing smaller things, right? It's about understanding the dynamics, how things actually happen in living systems.
Totally. Understanding those fundamental processes of life in a way we just couldn't before, from how drugs work maybe to how cells talk to each other. So yeah, prepare for some pretty surprising stuff.
Definitely.
Okay, so let's maybe set the stage first. We need to talk about the big hurdle the opponent microscopists have been battling for ages,
Yeah.
the diffraction limit. For anyone maybe not deep in optics, what exactly is this limit? Why is it such a problem?
That's absolutely the right place to start. Because the diffraction limit, it isn't just, technical inconvenience. It's a fundamental law of physics. It governs all traditional light microscopy. Basically, think about light as a wave. If you have two objects that are really close together, closer than about half the wavelength of the light, you're using their light waves kind of blur and overlap.
Ah, okay, so they just look like one blob.
Exactly. One indistinct blob. You lose the separation. And for the visible light we typically use, that limit is somewhere around 200 to 300 nanometers.
Which sounds tiny.
It sounds tiny, but here's the kicker. Think about biology. Many of the really critical players, individual proteins, tiny machines inside our cells, those little vesicles carrying cargo around, even where a drug molecule binds, they are often way, way smaller than 200 nanometers.
Oh, right, like tens of nanometers.
But we're even
Yeah,
smaller.
precisely. We're talking tens of nanometers, sometimes single nanometers. So if your microscope's limit is 200 nanometers and you're trying to see a 5 nanometer protein, well, you're kind of flying blind.
You know something's there, but not really what it looks like or who it's interacting with.
Exactly. You can't make out its shape, its partners, its subtle movements. It's like trying to read really fine print with seriously blurry glasses. You just can't resolve the detail.
So back to your dance analogy, instead of seeing individual dancers, we just see this hazy swirl, missing all the important steps, the interactions.
You've got it. You missed the crucial choreography of life.
And it's not just seeing the small stuff, is it? You mentioned movement. It's also about seeing things change fast. That's the other half of the challenge, absolutely. Which brings us up to the need for both speed and precision. High spatial precision, seeing small things clearly, and high temporal resolution, seeing fast changes accurately.
Because things in biology don't just sit still.
Not at all. Scientists need to understand how things move, interact, change, often on incredibly short timescales, milliseconds, even microseconds. Think about tracking, say, a single enzyme moving along DNA. That's a subdiffraction-sized object, and it's moving. Or observing those random stochastic flashes of single fluorescent molecules blinking on and off inside a cell.
Those blinks actually tell you things, right, about the molecule's environment.
They do. They contain information.
Yeah.
Or studying how cellular compartments self-organize thousands of molecules, constantly shifting, binding, unbinding. These are super dynamic processes. And traditional methods, well, they really struggle to capture the full picture. You often get a blurry average, or maybe just snapshots that miss the critical moments in between.
Like watching a movie with lots of missing frames. You get the gist, but miss the key action.
Exactly. You miss the split-second plays, the nuances of the movement.
Okay, so if these dynamic nanoscale events are so tricky, what were the tools people were using before these neuromorphic cameras came along? What were the limitations of those conventional cameras?
Right. So for a long time, the workhorses in this field have been things like EMCCDs, that's electron multiplying charge coupled devices, and SCMOS cameras, scientific complementary metal oxide semiconductor devices. And these cameras have been amazing tools, don't get me wrong. They really push the limits for low light imaging, for speed. EMCCDs, for example, are brilliant at amplifying really faint signals, taking just a few photons and making them visible.
Okay, so they're sensitive. What about SCMOS?
SCMOS cameras are often valued for their speed and large field of use. You can see a bigger area faster. Both have been really indispensable.
And how do they generally work? Like, what's the basic idea behind how they capture an image?
They both fundamentally rely on what's called an integrative approach.
Yeah.
It's basically like taking a regular photo or shooting video. The camera sensor collects photons, light particles, over a set amount of time.
And exposure time.
Exactly, an exposure or integration time. It gathers all the light hitting each pixel during that window and integrates it into one value for that pixel in the final frame. So you get a series of snapshots.
Like 30 pictures per second or whatever the frame rate is.
Precisely. Each frame represents the average of everything that happened during that specific integration period. Whether anything changed much or not, it captures the whole scene.
Okay, that sounds fine for everyday photos, but I can see how that might be a problem for the tiny, fast, flickering stuff we're talking about. What were the main drawbacks of that snapshot approach?
Yeah, the drawbacks become really significant for these fast biological events. First, that integration time. Once you set it for recording, you can't change it afterwards. You're locked in.
Okay, so you have to guess the right exposure beforehand.
Right. And that's a huge constraint. If you set it too long, you might blur out super fast events completely. Imagine a molecule blinking on and off really quickly within your exposure window. You might just see a faint streak or miss the off part entirely.
And if you set it too short.
Too short. And you might not collect enough photons, enough signal to even see what you're looking for, especially if it's dim. So you're constantly making this trade-off before you even begin.
Speed versus sensitivity.
Exactly. Then there's the bandwidth issue. These cameras, especially when recording sparse events like single molecule flashes, they still process and send data for every single pixel in every single frame.
Even if most of the pixels saw nothing.
Even if 99% of the frame is just background. This creates enormous amounts of data, much of it redundant, which means huge files, slow transfers, heavy post-processing, a big digital footprint burden.
Right. I can see that being a practical nightmare.
It can be. And finally, there's the signal-to-noise ratio, the SNR. Getting a good, clean signal above the background noise is absolutely critical for seeing clearly and locating things accurately. But achieving high SNR often means sacrificing speed, using longer integration times, or blasting the sample with more light, which isn't always biologically friendly. So again, you're stuck in this difficult compromise. See clearly or see quickly. It was really hard to get both at the extreme nanoscale.
Okay, that really lays out the challenges. So now enter the neuromorphic cameras. This is where things get different. Let's unpack this. How are they such a radical departure?
Absolutely. Neuromorphic cameras are a truly different breed of detector. And what's so cool is they're directly inspired by how our brains handle vision.
Okay, brain inspired. How so?
The key thing is, they don't capture frames like regular cameras, no snapshots. Instead, they operate on an event-driven principle. They sense change. Think about your own vision. Your brain isn't constantly taking full pictures. Your neurons fire mostly when something changes movement. A light flickers, something new appears. Static stuff gets filtered out.
So it's more efficient.
Way more efficient. Neuromorphic cameras mimic this. Each pixel acts like a tiny, independent neuron. It only generates an event, a digital spike, when the light intensity falling on it changes significantly.
Only when there's a change.
Only then. And it does this asynchronously, meaning each pixel acts on its own without waiting for a global clock signal. If nothing changes for a pixel, it stays silent. No data, minimal power.
Wow, okay. So it's not measuring brightness itself, but the rate of change in brightness. It's recording the dynamics directly.
You've got it. They encode the rate of change of intensity, not the absolute intensity level. And this leads to some really transformative features. First, because only pixels seeing changes report anything, you get what are called sparse measurements. The data stream is mostly empty, containing only the important change information. This drastically cuts down the amount of data compared to sending full frames.
That solves the data overload problem you mentioned.
Big time. And maybe most importantly for dynamics, they have incredibly high temporal resolution. We're talking microseconds, orders of magnitude faster than conventional frame-based cameras. They can pinpoint when an event happens with extreme precision.
So microseconds, that's capturing motion almost instantaneously, pixel by pixel. That sounds tailor-made for those biological processes. Why is this event-driven way so ideal for the stuff traditional cameras struggled with?
It really is the perfect match. Because they only report change, they excel exactly where traditional cameras fall short, which is often where the most interesting biology is happening. Take those blinking single molecules, the stochastic emission. A normal camera might blur several blinks together in one exposure.
Right.
A neuromorphic camera, it registers the precise microsecond time the molecule turns on and the precise time it turns off. You get this incredibly detailed, unfiltered timeline of its behavior.
Wow.
It's also amazing for things like watching neurotransmitters being released at a synapse or tracking a virus entering a cell. These things happen incredibly fast. NM cameras can capture those fleeting moments. They're perfect for anything where the action is intermittent, localized, and really, really fast. And, as we said, because they're only reporting activity, the data footprint is way smaller, orders of magnitude smaller.
Less storage, faster analysis. That's huge for scientists drowning in data.
Absolutely. It flips the script from record everything, filter later, to only record what's changing and matters right now.
Okay, the theory sounds amazing, like a paradigm shift. But how do they actually stack up in a real-world test against the old guard, like EMCCDs? Especially for that crucial nanoscale precision. How did the researchers in that Nature nanotechnology paper actually compare them?
Right. The proof is in the pudding, as they say.
Okay.
So to really test this, the researchers did a direct side-by-side comparison. They put their neuromorphic camera, which they just called NM, against a standard EMCCD called EM. Their test subject was 100 nanometer fluorescent particles, a pretty standard benchmark for nanoscale imaging.
Okay, small enough to be tricky, but bright enough to see.
Exactly. Now, for the EMCCD, they synchronized its picture taking with a pulsed laser, so the light blinked on and off and controlled high and low states. This gave them a very defined input for the EMCCD.
Got it. A controlled rhythm for the traditional camera.
Yep, but the neuromorphic camera, the NM, it just recorded its events completely asynchronously. It wasn't tied to the laser pulsing at all. It just reacted whenever it saw a change in light from the particles.
That difference right there feels important. One marching to a beat, the other just reacting naturally. This comparison must have been key.
Oh, absolutely. This side-by-side under controlled conditions was vital to fairly evaluate the pros and cons and really quantify the advantages of the NM approach. Using those 100 nanometer particles really tests the precision needed near the diffraction limit.
Okay, let's talk dynamic range then. How well can they capture both dim and bright things at once? And event fidelity. How faithfully do they record those quick flashes? What did they find with the EMCCDs?
So EMCCDs, as we mentioned, are great for ultra-low light because of that electron multiplication gain. They can really boost faint signals. However, the researchers found their dynamic range, the ability to handle dim and bright signals simultaneously, actually decreased rapidly as they turned down that EM gain.
Ah, so boosting the faint signals maybe came at the cost of handling bright ones well.
In a way, yes. And also, if they reduce the integration time to try and capture faster events, even at the same laser power, the dynamic range of the EMCCD also dropped. So you were often forced into a compromise, high sensitivity or fast capture. But getting both with great dynamic range was tough.
Like trying to photograph indoors with a bright window in the shot, hard to get detail in both the shadows and the highlights?
That's a perfect analogy.
Yeah.
Now, how did the neuromorphic camera do?
Yeah.
Especially since it doesn't have that built-in gain.
Yeah, how did it compare?
Well, this is where it got really interesting. Even without that EM gain, the NM camera achieved a dynamic range of 14 to 15 bits, which is actually comparable to the EMCCD running at a pretty decent gain setting, like 50 to 100.
So inherently quite good dynamic range then.
Yes, but here's the crucial part. They found that changing the laser switching speed didn't change the dynamic range of the NM camera at all.
Whoa. Okay, what does that tell us?
It strongly confirms that the NM camera is truly recording the instantaneous rate of change. It's reacting to how fast the light changes, not how much light accumulates over a fixed time window. Its performance isn't tied to an arbitrary integration period. It's event-driven.
It's genuinely reacting to the event itself. And you mentioned on and off events before. How does that play into this? That sounds unique.
It is absolutely unique to neuromorphic detection. The NM camera distinctly detects both on events, when the light intensity goes up, a positive change, and off events when the intensity goes down, a negative change. It sees both sides of the coin.
Whereas the traditional camera mostly just sees the on-in state, the bright flash.
Primarily, yes.
Mm.
The EMCCD typically captures the high-state signal, but the NM gives you the precise moment something appears and the precise moment it disappears or fades. It's a much richer picture of the dynamics.
That sounds incredibly useful.
It's more than useful. It unlocks amazing flexibility. This ability means researchers can essentially recreate events at different temporal windows, at user-defined frame rates, post-acquisition.
Wait, after the experiment is done?
Yes. You capture the raw stream of timed events once, then later on the computer, you can reconstruct that data as if you'd filmed it at 100 frames per second or 1,000 or whatever makes sense for your analysis.
That's incredible, like having a time machine for your data.
Kind of. It offers unbelievable flexibility. You can revisit the data, analyze it at different timescales, look for correlations you didn't expect, all from the original recording. Impossible with fixed-frame cameras. It's like having a master recording you can remix endlessly.
That flexibility alone sounds revolutionary. Okay, let's get to the heart of it. Signal-to-noise, SNR, and localization precision. Pinpointing those tiny dots. This is where things get really interesting, you said.
Absolutely, because ultimately, seeing clearly and knowing exactly where something is, that's the goal. Think about finding the center of that tiny blurry spot of light from a single molecule.
Like finding the exact center of a fuzzy star.
Good analogy. The computer algorithms used for this, centroid estimation algorithms, they need good signal-to-noise and good dynamic range to work well. The cleaner the signal, the less the noise, the more accurately they can calculate the precise center.
Makes sense. So how did NM and EM compare here, especially in tough, low-light situations?
Okay, here's where it gets really interesting. The findings were quite striking. At low light levels, noisy conditions like working with sensitive samples or fast-fading dyes, the neuromorphic camera actually outperformed the EMCCD.
Outperformed it, even though the NM camera was first gen and didn't have that signal boosting game.
Exactly, which is pretty remarkable. It suggests that the fundamental event-driven design, by inherently focusing only on the changing signal and ignoring static noise, gives it an edge in those challenging low SNR conditions.
Wow. So in the dim, difficult scenarios, the brain-inspired camera was better. That's huge for biological imaging. What about when the lights were bright, optimal conditions?
Well, even under high illumination, where EMCCDs are typically strongest, they hit a performance ceiling. The EMCCD's localization precision and its SNR basically saturated.
Meaning more light didn't make the picture any clearer or the location more precise after a certain point.
Correct. The benefits plateaued. It reached a limit in how much information it could effectively use. The neuromorphic camera, however, kept improving. Its SNR and localization precision continued to get better as the laser switching or illumination intensity increased. It seemed to have more capacity to leverage the extra information.
So it had more headroom to keep getting better.
It did. So to sum it up, NM's precision was comparable to the EMCCD at its best, low gain, high light. But crucially, it outperformed the EMCCD when comparing signal to noise in those noisier low-light conditions. That's a clear advantage where it often matters most in biology.
That's a really powerful demonstration. A different approach shines where the old ways struggle. Okay, building on that, let's talk about the headline achievement. Getting below the diffraction limit. How well did these NM cameras do?
This is the breakthrough, really. The study showed the NM camera could localize these nanoscale fluorescent objects below that diffraction limit, achieving a precision better than 20 nanometers.
20 nanometers? That's tiny, small enough to see what kind of things?
Small enough to precisely pinpoint individual protein complexes, maybe specific structures within mitochondria, or even single ribosomes inside a cell. And importantly, when tracking these things, it kept precision below 50 nanometers, but with millisecond temporal resolution.
Wow. That combination is key, isn't it? Super resolution in space and
And
time.
that's the holy grail, seeing tiny things clearly and seeing them move fast.
And you said the NM camera gets both on and OFF events. How does that specific capability lead to such a big jump in precision? What was missed by just seeing the ON state?
This is the core innovation for precision. Traditional cameras mostly see the on-end state, the bright flash, like just seeing the lightning. But the NM camera records both on and off events as these nonlinear detections proportional to the rate of change. So it captures the dynamics of appearance and disappearance. It sees the lightning strike and the precise moment and way the afterglow fades.
Okay. And these on and off events give different information.
They give complementary information. The researchers found that the probability density functions, or PDFs, basically maps, showing the most likely location of the particle derived from on-N events, PON, and off events, POFF, contain spatial information about the same molecule, but in a mutually exclusive manner.
Mutually exclusive, like two different perspectives on the same location.
Sort of. Think of it like having two slightly different, slightly blurry maps of the same tiny spot. One map comes from the light turning on, the other from it turning off. Each map gives you a unique clue about the exact center.
So what did they do with these two different maps? How did that lead to the breakthrough in pinning down the location?
This was the elegant part, the eureka moment. They tried combining the information from both maps. They found that mathematically combining the ON and OFF PDFs, either by subtracting one from the other, or even more powerfully, by multiplying them significantly improved the accuracy of finding the particle center, the centroid estimation. And the key finding was that the map created by multiplying the two, PON-A-POFF, was the most robust way to estimate the location. It gave a much sharper, more accurate pinpoint. The accuracy was more uniform in all directions, better isotropy.
Multiplying them. Okay, so using both the light appear signal and the light fade signal together gives a much sharper fix, like using two spotters to triangulate a position instead of just one.
That's a great way to think about it. And the results were dramatic. The localization precision they got when combining on-N and off-F information was roughly 2 times better than using only the on-N information, like traditional methods implicitly do.
Doubled the precision, just by using the off signal too. That's incredible.
It really is a monumental improvement. And they further boosted this by using smart computational tools, things like deep learning, convolutional neural networks, CNNs, the deep track algorithm to help process the event streams and find those centroids precisely. It's a marriage of clever hardware and clever software.
Wow. Double precision is huge. Now, looking ahead, the paper mentions something intriguing. The super Heisenberg limit. That sounds very futuristic. What is it and what could it mean for future cameras?
It does sound futuristic,
Mhm.
but it's grounded in quantum measurement ideas. Basically, the classical Heisenberg limit in imaging says your precision improves relatively slowly as you collect more photons, roughly like the square root of the number of photons. The super Heisenberg limit suggests that with advanced detection schemes, maybe like these nonlinear methods, you could potentially achieve precision that improves much faster with each photon detected, maybe even exponentially. It's about pushing past what was thought to be a fundamental physical limit for light microscopy.
So, potentially even more massive gains in precision down the line.
Potentially, yes. Still theoretical territory. But the paper speculates that the next generation of these neuromorphic devices, especially if they add some on-chip amplification, could improve localization by another 8 to 10 times.
8 to 10 times better. What would that mean in practice?
That could push precision into the sub-nanometer range, seeing details down to the level of small molecules, maybe even atomic scale interactions in some cases. That would utterly revolutionize our understanding of molecular binding, chemical reactions, you name it.
Mind-blowing potential. Okay, so these cameras aren't just about static precision, they're also changing how we see movement. Let's talk about diffusion, that random jiggling of particles. Why is that hard to study, and how does NM help?
Right, diffusion, or Brownian motion, is fundamental. It's how molecules move and find each other in solution, how proteins navigate the crowded cell interior. Think of sugar dissolving in tea random movement,
Right.
but accurately tracking this random walk, especially over different time scales and at the nanoscale, is really tough. The movements are erratic, fast, and tiny.
And traditional cameras struggle with that randomness and speed.
They do, but NM cameras, because they record those two events, the precise moments of change with microsecond timing are incredibly well suited for imaging particles that are just freely diffusing around. You get this raw, high-fidelity trace of every twist and turn.
An unfiltered view of the random walk. So, what did the researchers actually see when they used NM to look at diffusion compared to the EMCCD? Did it reveal something new?
It revealed something quite profound about the nature of that movement. When they looked at the EMCCD data taken at different frame rates, like 10, 20, or 50 frames per second, the calculated diffusion characteristics actually changed depending on the integration time used.
Meaning the camera itself was influencing the result.
It seemed that way. The integration time was essentially averaging out or smoothing over the finest details of the particle's path, making the diffusion look different depending on how long the snapshot was. It was imposing its own timescale on the measurement.
Giving a simplified, maybe misleading picture of the actual movement?
Exactly. But the neuromorphic data, even when they analyzed it later using different time windows from 10 meters up to 100 meters, they saw something striking. The diffusion appeared more fractalized as the sampling frequency got higher,
More fractalized, meaning
meaning
more.
more jagged, more intricate, more complex at finer timescales. As they zoomed in temporally with the NM data, they saw more and more detail in the particle's path, tiny jiggles and sharp turns that were completely invisible, smoothed over by the EMCCD's longer integration times.
Okay, let's use that scribble analogy again. The EMCCD saw the general curve of the scribble. The NM camera, especially sampling fast, saw all the tiny, sharp, jagged movements of the pen making the scribble. The true complexity.
That's a perfect way to put it. It revealed that molecular motion at these scales isn't smooth. It's incredibly complex and irregular, fractal-like. This jaggedness could be really important for how molecules actually encounter each other and react inside a crowded cell.
It changes the picture from a smooth dance to something much more erratic and detailed.
It does. And this ability demonstrates the advantage of NM over EM for studying fractalization, as the paper says. It lets researchers ask new questions. How does the cellular environment affect movement? Is it slower than expected, sublinear, normal, linear, or maybe even faster, superlinear, due to other factors? It opens up whole new ways to probe the physics of life inside a cell.
A much clearer, less averaged view of fundamental processes. And did the on-in and OFF events agree on this diffusion?
Yes. Importantly, the diffusion characteristics they measured were consistent between the on-N and OFF event streams from the NM camera. Both types of events provided the same reliable information about the particle's complex movement.
That's great validation. Wow, okay, we've covered a lot. It really feels like this neuromorphic localization microscopy, this brain-inspired, event-driven way of seeing, is a genuinely powerful new tool. It's not just seeing below the diffraction limit down to below 20 nanometers, but doing it while also capturing dynamics at millisecond speeds. It's
Yeah.
bringing that invisible high-speed world into focus.
It really is.
Yeah.
And like you said, it's more than just a technical step up. It's a conceptual shift. Capturing the rate of change using both on and off events to effectively double precision. It gives us completely new insights into things like fractal diffusion that were just hidden before. We're forced to rethink some basic assumptions about how things work at the molecular level.
And the future seems bright. Less data to handle, synergy with AI and machine learning. These cameras seem poised to, as the paper puts it, ameliorate the shortcomings of conventional imaging and open novel avenues. Basically, fix the old problems and show us totally new things.
Exactly. They're providing the right kind of tool, almost a more biologically natural tool, to observe the intricacies of life in space and time.
So connecting this to the bigger picture, maybe the final thought for you, our listener, is this. With these seeing by change cameras becoming available, what previously hidden biological secrets, what fundamental dances of life, what molecular interactions that we've misunderstood or never even seen will we finally be able to uncover and truly understand?
Yeah, the potential for discovery across medicine, biology, maybe even material science. It feels pretty boundless right now. What will we see next?